Hello, everyone. My name is Natasha Heins, and my talk is about utilizing leaves to quantify changes in insect herbivory across facies in Harvard Forest, Massachusetts. I'm a senior biology student conducting research under Dr. Ellen D. Carano and Lauren Azevedo Schmidt. My project is a piece of Lauren's dissertation work where she is working to better understand abiotic and biotic variables influencing the paleobotanical fossil record. The samples I analyzed for my WRSP work were collected in the summer of 2019 by Lauren and her undergraduate student. These leaves will eventually be compared to two other forests, the Smithsonian Research Station in Edgewater, Maryland, and La Selva, Costa Rica. However, the two other sampling sites are outside of the scope of this project. A total of 4,115 leaves were analyzed from the Harvard forest and what I will be talking about today. Harvard forest is a temperate forest that is denoted by the star in the right-hand picture. It was placed here as its average temperature is about 8 degrees Celsius. It also has an annual precipitation of 110 centimeters. The leaves were collected from 4 different facies. The term facies is a geological term and can be defined as the characteristics of a rock or sediment unit that reflects its environment of deposition and allows it to be distinguished from rock or sediment deposited in an adjacent environment. In this context, they can be thought of as various environments. A swamp, a small tributary, which is a small reach of river that feeds into a larger body of water, a dynamic river, and an upland were sampled. The maps shown here show the relative position of Harvard Forest in panel A and B is a detailed map showing the temporal span of each location. Within each facies, quarries or replicate sampling sites were sampled to account for spatial variability. The trophic levels as shown here are important for understanding how ecosystems function in the past and present environments. Plants are primary producers with herbivorous insects acting as the primary consumers of plant material. As plants make up the base of the trophic pyramid, they control the overall health of the higher levels of the pyramid. By understanding how they are impacted by abiotic variables, which are environmental changes, and biotic variables, which can be insects and other animal interactions, we can better understand ecosystem fitness. Leaves in both modern and fossil sites allow us to analyze insects based on their feeding patterns preserved on leaves because insect body fossils are less abundant. By conducting research on leaves and the insect feeding preserved on them, we can look at how plant and insect communities change across various facies. Future work is being done on understanding the importance of abiotic variables on plant-insect communities, but that is outside of the scope of this talk. However, this work is important because as human-driven anthropogenic climate change continues to change our Earth, we need to understand these trophic levels because humans are a part of trophic pyramid and are heavily reliant on plant species. Humans are approximately at 2.2, which is the same as pigs within the trophic pyramid. All the facies we sampled for the study are abundant in the fossil record, allowing us to compare our findings to fossil localities in the future. As leaves are preserved in wet environments such as rivers, lakes, swamps, and ponds, we sampled them in modern environments to allow them for future comparisons. However, since leaves are preserved in wet environments, preservation of leaves is impacted by various types of transport and important to incorporate into studies. My contribution to this project was to photograph and measure various leaf traits on all 4,115 leaves. The leaf traits in insect herbivory measured were insect damage, leaf area damaged, petiole width, and leaf mass per area, also known as LMA. Damage was measured on the adaxial, which is the top of the leaf, and abaxial, or the underside of the leaves. These are shown in the pictures on the right. The leaf area was measured to compute the percent area damage or how much of the leaf has been eaten by insect herbivores. Insect feeding changes can be due to environmental changes such as temperature and leaf nutrition, but those were not measured here, however, they will be incorporated into the overall dissertation project that Lauren is currently working on. Petiole width is directly correlated to the mass of a leaf. This means that the larger the petiole, the bigger the leaf. Petiole width is important because leaf mass cannot be directly measured on fossils because a fossil leaf is just an impression and a 3D measurement cannot be made. The petiole is this part right here in case you need a reference. One hypothesis that we have is that LMA will be higher at the upland site rather than the dynamic river within the 1903 quarry, as they will be less destroyed. Using the Insect Damage Guide by Conrad Leberindera et al., Lauren quantifies the insect damage prior to me photographing and measuring the damage. Insect damage is broken into functional feeding groups and damage types. The circle above represents the functional feeding group, while the following images with the DT denotations are the damage types that make up the functional feeding groups. There are 7 functional feeding groups: whole feeding, skeletonization, surface feeding, galling, margin feeding, piercing and sucking, and mining. Various insects can make a variety of damage, but damage type diversity has been shown to correlate to living insects community and diversity. Lastly, the insect damage is distinct compared to a tear or a hole in a leaf, not produced by an insect because of the reaction rim, which is a thickened leaf material preserved on modern and fossil leaves. Without the reaction rim, we cannot confidently assign it as insect herbivory. An example of a reaction rim is up here in the left-hand corner, as I am showing. Our hypotheses for insect herbivory at Harvard Forest are that there would be changes in insect herbivory across facies due to various types of transport. We also expected more insect herbivory at the upland site because of less transport and preservational bias, and assumed higher leaf area. Now, let's look at our results. Rarefaction curves were produced to analyze the insect damage and tree species richness across all facies within Harvard Forest. The color codes used here are consistent throughout the talk, with blue being the swamp, green being the small tributary, yellow being the dynamic river, and the pink being the upland facies. The x-axis for both plots shows the number of individual leaves sampled. The y-axis is either the number of damage types for panel A or the number of tree species for panel B. Both panels show that we did not saturate our sampling because none of the lines reach a saturated or asymptotic state. Although we did not achieve this with our samples, we still see in panel A that there are distinct differences in insect richness across facies. However, panel B shows that given a larger number of samples, the dynamic river would have surpassed the small tributary for tree species richness. The least diverse sites are the upland and the swamp. In these graphs, panel A quantifies the changes in average total damage across the facies. We see that the upland site is significantly different from the others, and so is the small tributary, but the two are different from one another, denoted by the A and B. Additionally, the upland site has the highest mean total damage when compared to all the other facies. This is interesting, as even though it has the least number of leaves, it had the most damage. We developed a hypothesis that we expected the upland site to have more damage, as there is not preservation bias present. This means that the leaves are dry and not being transported within the environment. Lastly, the swamp and dynamic river group together and are statistically different than the small tributary and the upland. In panel B, it quantifies the changes in leaf area across the facies. The upland site and the small tributary were significantly the same, denoted by A, while the swamp and dynamic river were statistically the same, denoted by B. We expected the upland site to have the most total damage and larger leaves because of the lack of transport, which biases towards smaller leaves. However, what is interesting is that the leaf area of the upland and the small tributary are statistically the same, but they differ in average total damage as shown in panel A. Although the swamp also undergoes relatively low to no transport like the upland facies, swamps are notoriously patchy landscapes. Panel C quantifies the average percent area damage or the amount of leaf area an insect has eaten, and we see no statistical differences between the facies. Although there were differences in average total damage as shown in panel A, the overall amount of percentage of leaf tissue consumed was similar across the facies as shown here in panel C. Lastly, in panel D, it shows that the leaf mass area across the facies. Here we see a similar pattern as with panel C, whereas there is no statistical difference across the facies. However, unlike other panels, the swamp has the highest LMA when compared to the other facies. This isn't statistically significant difference, however. Further work will look at the spread in LMA within the facies to see if it is if there will be more informative of the variability. In summary of the previous plot, in panel A, the upland site is significantly different from the others, and so is the small tributary site. However, they are different from one another. Also, the upland site has the most total damage. In panel B, the leaf areas are statistically similar for the upland and small tributary sites, while the small and dynamic rivers are statistically similar. Although the upland leaf area is the same as the small tributary, the average values are the highest. In panel C, there is no change in percent area damage across the facies. And in panel D, there is no change in LMA across facies. Due to differences in sample size correlated to the petiole preservation, it's important to highlight that LMA values were quantified on 60.46% of the leaves for the swamp, 54.16 for the small tributary, 63.4% for the dynamic river, and 95.88 for the upland. Now moving on to NMDSs. An NMDS is a non-metric multidimensional scaling. It is a type of ordination which compares the similarity or dissimilarity of a matrix. Here, we are looking at how insect functional feeding groups and facies plot in multi-dimensional space. We can see that all facies plot relatively close together, which means that all the quarries that make up a facies are relatively the same in their insect herbivory. The small tributary points plot the closest together and therefore are the most similar. The spread in the swamp samples, denoted by the blue triangles, is likely indicative of the patchy nature of swampy landscapes. However, the spread in the dynamic river points need further investigation, but are likely due to a sampling bias in the collection methods. The swamp facies is dominated by galling insects, whereas the dynamic river is influenced by surface feeding, skeletonization, and hole feeding. The small tributary is influenced by margin feeding and the upland plots in its own space, but is closest to mining insects, which is likely control in its relative position. In conclusion, we know that insect herbivory within Harvard Forest is much more diverse than assumed prior to this study. This is likely due to the anthropogenic climate change and increasing Massachusetts temperatures. Also, insect and plant richness do not neatly correlate to one another. Facies differ in average total damage in leaf area, but not in average percent damage or leaf mass per area. The NMDS shows that which functional feeding groups influence the position of the facies, with all quarries plotting within the similar space of one another. With this future research will compare the other forests previously mentioned to these findings to better understand the influence of abiotic and biotic factors on plant and insect interactions. I just wanted to thank Dr. Crate for this symposium and running WRSP. I also want to thank Dr. Ellen Carano and Lauren Azevedo Schmidt for mentoring me and teaching me so much about research and science, along with how important it is to be a woman in STEM. and I want you guys to have a great day and thank you for your time.